PCR major trial between IOS and OA plus, and there's no significant difference between
no
IOA,
difference.
no difference.
I see.
The other question I always have is that the activation status of the macrophage in RA versus RA, because in general it can be classified into M1 and M2, and M2 can be classified into 2A, 2B, and 2C. I'm not sure this system can be applied into RA or not, and this.
Hold this.
What?
Hold the camera.
Hold the camera.
Yeah. Okay, so yeah, the differentiation state of macrophage is critical because trail kills which one?
I don't know. I'm not very sure.
What was it? It was
sensitive to trial.
Not all of them though.
Probably some of them.
I think it was a CD11B dull. High. Okay.
Yeah,
so
high. It's
goes like
sensitive.
this. Sensitive. And now there are also these, because actually we actually were very familiar with this. This is PDCs. We like those. But anyway, these are CD11C, CD11B high. We don't know what that means either, but there actually was one other factor, CD11B high. And what else was it? That was what's called the natural macrophage. Remember what that marker was? What was that marker?
I know we did some LY.
6C.
But there was a natural. Anyway, the problem was the ones that were killed were what was thought to be the anti-inflammatory macrophages, as I recall.
Like NDSC.
Huh?
NDSC, CD11B positive and GR1B positive.
Oh yeah, GR1. Yeah. So you did the CD11B, GR1. There was actually this one. I mean, there was actually kind of a mix of these, but I think there was a GR1 positive, which was the non-inflammatory one. I forget how they determined it.
Suppressor.
Yeah, suppressor macrophages. Maybe you can learn something from that. But that's one thing. So yeah, so as opposed to being the activated macrophage, okay, CD11B, that sounds good. But GR1 positive, you always run into this problem that maybe the cell type that you want to kill has been selected for. We have that with the transgenic mouse.
This population can belong to one of these regulatory macrophage.
Yeah, but why would the regulatory one? Is that artificial? That's what we
probably
can.
Belong to regulatory. I'm not very sure.
So but what was the first thing you said about the macrophage? What was this over here? You said what was it that was similar between RA and OA?
The trial is the same.
Trail expression.
We measure the trial measured between RA and OA, fibroblast, and there was no difference.
Oh, this is fibroblast.
Yeah, fibroblast, and also maybe tissue. There's no.
Yeah, you need the tissue.
We take a tissue too.
What about your little chunks?
Haven't done it. And from the frozen tissue, there's no difference.
No difference, right? I remember that was because the fibroblasts, of course, they may or may not produce trail, so it's not the proper thing to look at. But the tissue in the synovium and the surgical or the time of surgery or what you're getting now with the synovial fluid, that might be the right thing to look at. So, what else? What else about this?
Can you send more?
Yeah. What
to the
is this first off, is this process ongoing in vivo? That'd be one thing.
Yeah.
I mean, because you see 1 million diagrams of this NK cells killing tumor, through trail and through other things. That's you copy and paste it, everybody pastes it in the paper. Probably did anybody ever show that even? But it's hard to show this. One thing is DR5, even though it's expressed everywhere, the trail is not expressed everywhere, but the sensitivity to apoptosis is not high. You know, even Tangzhou's antibody, his good antibody, you know, Tra8, it's crossing so well, it's not toxic in humans. Now, why is it that trail doesn't kill anything? You know, why is it you signal trail and nothing happens?
Yeah, I also tend to think that fibroblasts from RA is more sensitive to trail compared to those from OA.
Yeah, you seen that or not seen it?
I think
Yeah,
so
right,
it's
they're-
kind of comparable to the sensitive compared to the China. So I don't know why people always say that RA is resistant to apoptosis because those are more sensitive to China. I don't
Yeah.
know why.
Yeah, well, with Tangzhou's they didn't say it was more resistant. Oh, to trail or
to
DR5?
trail, to trail.
To trail. Oh, but you found out it's sensitive to
It's
trail.
more sensitive compared to OA.
Well, they studied.
Overall, it's more sensitive.
Well, they studied the passage ones, you know. Nobody's done it, you know, it's people miscopy other people, mistakes. But yeah, I mean, it could be that what you have is a real thing, you know, you're the one, you know, you found out, you know, you read reports and there's all these anti-apoptosis molecules are high in RA, right?
That don't go to the, what you just say, don't go to YouTube.
Yeah, okay, no, I mean, well, no, I mean, I'm saying that people study in vitro systems may not be what's going on in vivo. That's what I was talking about, remember I was saying, what's going on in vivo? That at least is a first clue. And then what goes on in what goes on in the petri dish or what goes on in samples that are available, that's the second level. But you know, that's what we, you know, so we got to think about what goes on in reality, and does it even occur? You know, now you can force it to, but it'd be better if it did occur. I mean, I'm trying to think of even what is the target in vivo? Is it, you know, we keep thinking of killing the synovial fibroblasts, but maybe the macrophage is the real target, maybe even in a tumor. The real target is the macrophage, you know, because we just don't know, but you know, it does kill macrophages, and you start to look at it and then you get this conundrum, it doesn't kill the macrophage. But, so there's so much that we don't know about this, and you know, they get bits and pieces from different things. So much we don't know about what produces trail, but I think it's in K cells. What the target of trail is it the tumor, the fibroblast, or is it the inflammatory cell, the macrophage? And then, you know, then there's the regulation of the signaling, and what is it there? Well, one thing is we haven't looked at the regulation of the macrophage. And then there's this, then there's the peroxidase, these sorts of things. They say the inflammatory cells produce factors, and they actually affect the apoptosis of this cell because of these factors. You know, so again, it leaves this cell off the hook. But we're saying, we're writing a whole paper that macrophages are the good target. You know, DR5 can kill macrophages. And we didn't really want that result, remember? I mean, I remember sitting there thinking, you know, okay, we struggle, and tried to hide and move those CD68 positive cells off the picture. Was 68 or 64?
68.
Yeah, 68. Trying to move those cells off the picture, and everybody said, you know, get rid of them. But finally they're there, and then the apoptosis, and then I guess we decided, well, let's make macrophages the target. And then we have the lysine Cre result that, you know, in those mice where you overexpress human DR5 on a mouse macrophage, monocyte macrophage lineage, that's when you can prevent arthritis. So maybe it really is inflammatory. Maybe it's an anti-inflammatory molecule. You know, that would start to fit with, you know, because inflammation is generally bad in general. I mean, it's good, but you got to regulate, you got to down-modulate. So with it, like with the T cell and the fast ligand, that's what that finally turned into, down-regulating T cells. Maybe over here, it's really the main function of DR5 is to down-regulate inflammation through macrophages. You know, and it makes sense too, because the innate immune response. NK cell, innate. Macrophage, innate. They're working together. Tumors, synovial fibroblasts, brain cells, liver cells, they're part of the body. But inflammation, so we've been taking DR5 away from the inflammatory route, which fast ligand turns into putting it on other cells. They're all on all the cells. But maybe just like fast, it regulates innate immune responses, more than macrophages even. And NK cells